Hey folks, it's me, Mr. Lang, and I'm here to talk to you today about diffusion and osmosis as a part of homeostasis. So we're going to talk about how stuff gets into and out of cells, and we're going to be talking mostly today about water, which is osmosis. That would be the diffusion of water through a membrane. Um, and we're also going to talk about how stuff, solid stuff like sugars or salts or proteins, things like that, might be able to get in and out of cells as well. So, before we get started, we have to talk about what a solution is. And I know for your whole life you've probably heard um, the term solution. It's a bleach solution. It's a cleaning solution. It's some kind of a solution. Well, every time you hear solution, from now on, I want you to think, oh, that's a solution. That means it's made of two things. It's made of a solute and it's made of a solvent. The solute is the stuff that gets dissolved. The solvent is the stuff that does the dissolving. So, for example, if I take sugar and I put it in water, I've made sugar water. Sugar water is a solution. The solute is sugar, the solvent is water. So you want to be thinking about solutions as having two parts, particularly as we go through this part of our class. So if we look at the example down here, we've got two different solutions. We have a concentrated blue solution and we have a dilute blue solution. So if we look here, we can see, oh, make that go away. We can see that the blue particles that we've got here are the solute, that's the solid. In this case, it's like a blue dye of some sort, probably. And the solvent, that's this purple thing here. If this is just, maybe this is just food dye and water. So the blue is the food dye, and the pinkish stuff would be representing the water. So in this case, we have lots of solute and not lots of solvent. And just the opposite over here. We added some more water to it, we diluted it, and so now we have fewer solute particles in there and it's more dilute. I think that's pretty self-explanatory, but it gets tougher, so keep writing. Also, as you're taking notes today, make sure that you're stopping the video if you need to. Don't miss anything because we're going to use all of the terms, solute, solvent, solution, all those things, and everything else I'm going to talk about are going to become very important as we go into next week. So, this is a term that we're going to use a lot in the next couple of weeks. So the concentration gradient is really, really important. Um, it doesn't physically exist, but it's sort of a theoretical construct that we use to explain how stuff moves in a solution. Um, so basically, stuff is always going to want to go from high concentration to low concentration until there's an equal amount of it through the entire solution. So, for example, if I was to have a really big beaker and I dripped some food coloring into it, you could watch that food coloring spread from that first little ball of food coloring. It would spread out slowly and over time because of water molecules moving in that solution and bumping into each other and bumping into the dye particles. Eventually that whole beaker is just going to be the exact same color. So we'd be going from a high concentration of dye to a low concentration of dye. And let's just think about what that means for a minute. Things want to diffuse down the gradient. That doesn't take any energy. It's really, really easy. What if I wanted to get that dye back? That would be really, really hard to do. Wouldn't be hard to do. I could just boil the water out, but it would take a lot of energy to go back up the concentration gradient. So hopefully that makes sense. So the concentration gradient, things just want to go from high concentration to low concentration. They go from organized to disorganized. That's the way that the universe works. Okay, moving along. Um, Also important for you to realize that the rate of diffusion, so how fast something spreads out, how fast that dye spreads out through the beaker, has a lot to do with how steep the concentration gradient is. So if I've got a lot of dye concentrated in a small area, it's going to spread out pretty quickly. But if I have some dye already in the water and I just add some more, it's actually going to spread out more slowly because the concentration gradient isn't as steep. Two terms that are really, really important. So I'm going to move myself so you can see them all. So diffusion is the movement of a substance from an area of high concentration to an area of lower concentration. That could be any substance: sugar, salt, protein, people, dye, smoke, um, anything. Smell. So if I was peeling an orange in the corner of the classroom, that would slowly work its way across the room and everyone would be able to smell it because it's a pretty pungent smell. Or if I opened up a beaker of vinegar and started pouring it back and forth in between beakers or something like that. Osmosis, that's the movement of water from an area of high concentration to an area of lower concentration. But it has to be through a semi-permeable membrane. So it has to be going through a plasma or a cell membrane in order for that to be called osmosis. We're actually going to do that in class, and you're also going to do a thought experiment, sorry, not thought, a virtual experiment with that online. So, so that's the difference between diffusion and osmosis. Osmosis is diffusion, but it's the diffusion of water through a membrane. And it has to be water, and it has to be going through a membrane, or it's not osmosis. If it's just something going from high concentration to low concentration, that's diffusion. Um, but so osmosis is a special case of diffusion. Um, so here's an example of osmosis. Here is my semi or selectively permeable membrane. On this side, we have some sugar water. On this side, we have some pure water. Well, this membrane is permeable to water, but not to sugar. Where's the water going to go based on what you know about diffusion and osmosis? Is the water going to want to leave the sugary side, or is it going to want to go to dilute the sugary side? Think about that for just a second. Realize that sugar can't cross this, but water can cross this. So the water is going to end up crossing the membrane, and the level on this side will go up. So we've got the diffusion of water across the semi-permeable membrane. What do we call that? That's right, we call it osmosis. And that's going to dilute this as far as it possibly can, until it gets to a point where both sides are in equilibrium, which would never happen in this case because we can't dilute this side as much as this side is diluted because this is pure water. So water will keep moving over to that side until there's no water on this side. And that's how osmosis will work in this case. Three terms that are really, really important when we're talking about this sort of stuff are isotonic, hypotonic, and hypertonic. So those are three different kinds of solutions that you can have. The example that they're showing here is a red blood cell. I know it doesn't look red. It looks like a brown blood cell, but that's okay. Maybe it's not doing so well. It's just about dead or something. But so we've got this red blood cell here, and you can see that there's the exact same concentration of solutes. Remember those dissolved solid things? The exact same concentration of dissolved solutes inside and outside the cell. Well, what's going to happen to water in that case? No net movement of water. Water is going to come in and water is going to go out, but it's going to be going in and coming out at the same rate because the concentration of dissolved solutes is the same inside as it is outside. That's isotonic, or the same tonicity. Hypotonic, what hypotonic means is you've got more pure water outside the cell, if you're talking about the solution, than you have inside the cell. So here we've got our solute particles here, just like we had over here, and the little dots represent water. So we have more dissolved solutes in the cell. Well, if we have more dissolved solutes in the cell, that means we have more pure water outside, and the water is going to want to go in. It's not going to want to go in. It doesn't want to do anything, but chemically it's going to go in. It's going to, through the process of osmosis, it's going to go through this cell membrane, and it's going to diffuse through a cell membrane, so we call it osmosis, and that's going to cause the cell to swell, and that's called cytolysis. Third type of solution that we could have. we could have more dissolved solutes outside of the cell than we have inside of the cell. If I have more dissolved solutes outside of the cell, that means I have more pure water inside. The water needs to go to try and reach equilibrium, so it's going to leave the cell and it's going to try to dilute the area outside of the cell. That's not going to happen. The cell is going to shrink. That's called plasmolysis. So in an isotonic situation, we have the same amount of dissolved solute inside and outside. In a hypotonic solution, we have more dissolved solutes inside the cell and fewer dissolved solutes in the solution. And in a hypertonic solution, we have more dissolved solutes outside the cell and fewer inside the cell. Well, let's try this out real quickly. So think about isotonic, hypotonic, hypertonic. So I've got 3 examples here for you. Is the solution hypotonic, hypertonic, or isotonic? Let's look. So here's the example. So here we've got a cell being represented by this brown colored circle. Here's the cell or plasma membrane, and here's the outside of the cell. So this is what we're going to be calling the solution. So inside and outside, do we have more or less solute, these little green circles? More or less or the same? What do we have here? Think about that for a minute. If you're thinking that it looks like it's pretty much the same, what term from this page should you use to describe it? You should call it isotonic. Okay, so is the solution hypotonic, hypertonic, or isotonic? Well, it is isotonic. So that is the term that we would use to describe it. This whole system is already at equilibrium. We have the same amount of dissolved solutes inside and outside of the cell. So water is going to have a net movement of 0. There's not going to be water. There's going to be water going in and out, but it's going to be going in and out at the same rate. Example 2. So here is example 2. Describe this solution. Think about the terms. What term describes this? Do I have more or less dissolved solutes in the solution? If I have more, I call it hypertonic because hyper means high level. If I have less, I call it hypotonic. That means low level. Which one is this? Do I have more or less? In this case, I've got less solutes out here, so I would call this hypotonic. I bet you can't guess what the answer to the last one is. Is this solution hypotonic, hypertonic, or isotonic? Well, are there more dissolved solutes out here than there are in the cell, or are there less dissolved solutes out here than there are in the cell? If there's more dissolved solutes out here, it's hypertonic. So those are our 3 examples. You can watch the last little segment again if you had any trouble understanding any part of it. But realize that these 3 terms, along with diffusion, osmosis, and concentration gradient, are going to be used a lot over the course of the next couple of weeks. So you definitely want to get a really good grip on what they all mean. If you have any questions, make sure you stop by and see me. We're going to be working with these terms, like I said, quite a bit. So make sure that you ask if you have questions. Take care. Hope this was helpful.